Okay, good afternoon. I'd like to welcome everyone to today's Walls Lecture, and I'm very pleased to introduce our distinguished speaker, Dr. Joe Takahashi, who will be telling us a fascinating story about the discovery of our internal timekeeping system, the circadian clock. This molecular timepiece is fundamental in synchronizing organisms with their environment and in directing internal physiological and molecular processes, including gene expression, the cell cycle, and epigenetic modifications. Circadian clock is emerging as a new frontier for understanding health and disease. Now, Dr. Takahashi is a world-renowned pioneer in the molecular and genetic discovery of circadian clock genes and their functions. He championed the use of forward genetics to discover the first mammalian circadian clock gene, which was called clock. Dr. Takahashi is at the forefront of new clock gene discovery in defining an intriguing transcriptional translational feedback loop of circadian clock genes that's both self-sustaining and cell-autonomous, and in the groundbreaking discovery that functional clock gene programs are present in virtually all tissues and all cells throughout the body. Dr. Takahashi received his bachelor's degree in biology from Swarthmore College in Pennsylvania. He completed his PhD work at the University of Oregon, and then came to the NIH for 2 years as a pharmacology research associate at the National Institute of Mental Health. From 1983 to 2009, he was on faculty at Northwestern University in Chicago in the Department of Neurobiology and Physiology, where he led a state-of-the-art circadian research program. During this time, Dr. Takahashi served as the associate chair of the Department of Neurobiology and Physiology, was awarded the Walter and Mary Elizabeth Glass Endowed Chair in the Life Sciences, directed the Center for Functional Genomics, was elected a Fellow of the American Academy of Arts and Sciences, was elected to the National Academy of Sciences, and became an investigator in the Howard Hughes Medical Institute. Dr. Takahashi is currently chair of the Department of Neuroscience at UT Southwestern Medical Center, where he holds the Lloyd B. Sand Distinguished Chair in Neuroscience and remains a Howard Hughes investigator. Dr. Takahashi has published over 200 peer-reviewed articles and has an extensive funding record, including an NIH Merit Award from 1987 to 1997. He's received over 35 major awards and honors and has served on nearly 40 advisory and editorial boards, including at the NIH, the Genomics Research Institute for the Novartis Foundation, the Allen Institute for Brain Science, and the Max Planck Institute for Biophysical Chemistry. He's presented over 300 invited lectures, including a Nobel Forum Research Lecture in 2000. Dr. Takahashi was a co-founder of a biotechnology discovery company that investigated sleep and wakefulness neurobiology pharmaceuticals, and he just completed a tenure as a president for the Society for Research on Biological Rhythms. So these are only a few of his long lists of accomplishments and honors. His lecture today is titled Clock Genes and Clock Cells, A New View. And I'm very pleased to turn over the microphone to Dr. Takahashi.
Thank you very much, Aaron. It's a real pleasure to be back in Bethesda. I actually grew up in Bethesda and Rockville when I was in elementary, junior high, and high school. So these are my old stomping grounds, so it's always nice to come back to the NIH. So I want to thank Aaron for embarrassing me in his introduction. What I'd like to do is to first give you sort of a historical view of how we got into studying clock genes. And then I'd like to turn to a few more recent examples of how we study clock genes and what their functions might be and how the system is organized. And so today, as a neuroscientist, it's a very exciting period because our ultimate goal is really to understand behavior and disorders of behavior in the brain. And we're, as you know, undergoing a revolution in both genetics and genomics. And so the combination of neuroscience and genetic approaches has really opened up a number of approaches to understanding behavior. So our interests have been to identify genes that ultimately control behaviors and behavioral disorders. And so what I want to do today is to try to show you at an example, the circadian system, in which we can work at all these levels of organization, starting with behavior, working backwards to identify genes, and then to use those genes to study how circadian rhythms are generated in cells and in circuits. And one of the themes of today's talk is really that the cell is really the fundamental unit of the clock in the body, but that the organization of these cells into circuits actually transforms this information to control behavior. And so it is important, if we want to ultimately make links from genes to behavior, to understand these different levels of organization. And the circadian system is really a beautiful example of this, where, as I'll show you, we have identified a number of clock genes involved in this pathway. Cells and circuits, primarily in the suprachiasmatic nucleus of the brain, are involved in generating circadian behavioral rhythms, but also to regulate physiology and metabolism. And alterations in physiology and metabolism ultimately could lead to or have impact on disease and pathology. So in humans, sleep-wake is really the most obvious rhythm for us, and it's clock-controlled. This is a very old experiment done in the 1970s where a medical student volunteered to go into isolation in an underground bunker at a Max Planck Institute in Erlangen-Ondex. And so he's living in an apartment with the door open, and this is a wake-sleep plot. He's awake when it's blue, he's sleeping when it's yellow, and the arrowhead shows the low point of his body temperature rhythm. And each line is a day. He's living in the apartment with the door open until this point. Then they close the door and isolate him from all temporal cues. No clocks, no TV, no radio. He has 24-hour room service. And what you see is that his sleep-wake pattern continues, but that the timing of it drifts to later and later times of day. Okay, so that by this day here, he's waking up at midnight instead of 8 o'clock in the morning here at the beginning. Okay? On this day, the door to the apartment was opened again, but he continued to live in this apartment. And you see a second feature of these rhythms, and that is he goes back to a regular 8 a.m. wake-up time and going to sleep around midnight. And his period length is now 24 hours. It's synchronized to the environmental cycle. So rhythms are generated internally by a clock in our brain, but the environment plays an important role in synchronizing our rhythms, both to control the period length, to make it 24 hours, but also, importantly, to control the phase. So we as humans are diurnal, and you see that he goes back to being diurnal, waking up at 8 a.m. Now there's a curious feature in this record here. Why is he over here at 8 a.m. on this cycle as opposed to 8 a.m. over here? It turned out, while he was in isolation, since he was in there a long time, he actually lost an entire day. He went through one fewer biological cycles than the number of real days because he's waking up more than an hour later each day. Okay? So those are the features of rhythms. In people, light is the major synchronizer of our rhythms. There's a specialized set of photoreceptors, a new class of photoreceptors in retinal ganglion cells that mediate this information. They project into the hypothalamus, into these two yellow wing-like structures, which are called the suprachiasmatic nuclei. And within each nucleus are about 10,000 neurons, each of which can generate circadian oscillations. So even in mammals, the fundamental unit of rhythm generation is the single cell. The circadian clock is cell autonomous. So the The modern era of circadian rhythms was really started by these two individuals, Ron Konopka and the late Seymour Benzer at Caltech. And this is a picture taken in about 2000, much later than Ron's graduate career. But in the 70s, Ron published a classic paper in which he isolated 3 mutations in flies that either shortened, lengthened, or abolished the rhythm. And these 3 mutant alleles mapped to a single locus that they named the period locus. That was in 71. It took 13 years to clone the gene. That was done in 1984 by Michael Young at Rockefeller and Mike Rosbash and Jeff Hall at Brandeis. But the cloning of PER was not very informative because PER is a very unusual protein. It had very few obvious motifs. And so it wasn't until much later, around 1988 or 90, that the Brandeis group realized that the PER protein and RNA was cycling in the heads of flies. And this led to the idea that PER might be involved in some kind of autoregulatory feedback loop. Now, during this time, everybody in the field tried to clone PER homologs in mouse or human, and we all failed, and that's because the nucleotide sequence is highly divergent. And so in the early 90s, we began to learn that you could do forward genetics in the mouse, and we collaborated with Bill Dove, then we're still working at the University of Wisconsin at Madison, who was using ENU, or ethyl-N-nitrosourea mutagenesis in mice, to isolate mutations in a biochemical enzyme. And so with Bill, we collaborated. They did the mutagenesis in Madison, then shipped down their negative mice to Evanston at Northwestern, where we screened them for behavioral phenotypes. So this is an actogram of a mouse. It's double plotted. The dark shows when the mouse is running, and the light bar up here shows the dark light cycle. Mouse runs during the dark. It's on a light cycle for the first 2 weeks or so. Then it's transferred to constant darkness, or DD, here. And then we see the expression of its endogenous period, which in the case of the mouse is shorter than 24 hours. In mice, the period is extremely tightly regulated. And so in the first 300 mice screened by Martha Vitaterna in my lab, this is the distribution of the period lengths of those mice. Average of 23.7 hours with a standard deviation of only 12 minutes. And in that first screen, Martha isolated this mouse, which has about a 25-hour period to its clock, much more like human circadian rhythms. Okay. This mouse is shown here, and it is clearly divergent from wild-type mice. We subsequently showed it was caused by a single autosomal mutation, and in intercrosses where we took presumptive heterozygous mice to produce our first homozygous mouse. This is actually the first homozygous mutant clock mouse that we saw. It's pretty normal on a light cycle, but in darkness, it has a 28-hour clock, 4 hours longer than normal. And then the rhythm disappears or fragments in constant darkness. The heterozygotes have a 1 hour longer clock, and then the wild type has a normal period. And in fact, this was actually Martha's first litter, which the Mendelian prediction would be a 1 to 2 to 1 ratio, wild type heterozygotes to homozygotes, and that's exactly what she got. So after characterizing the mutant mouse, we were able to take advantage of the beginnings of the Human Genome Project and use positional cloning to ultimately identify the clock gene. This slide here summarizes about 30 person-years of effort in my lab, working as a team for 3 years to clone the clock gene, which turned out to be incredibly interesting. It was a novel member of the basic helix-loop helix pass family of transcription factors. It shared a motif with the original period protein, the so-called PAS domain, which we now know is a dimerization interface. But importantly, it also had a clear DNA binding motif, a BHLH, and a glutamine-rich C-terminus, which suggested it might be an activator. And so using clock, we are able to then identify BMAL1 as its heterodimeric partner using use2 hybrid screening with Chuck Weitz at Harvard. And ultimately, we showed that clock and BMAL can activate a number of genes, the period genes in mice, as well as another class of gene called the cryptochrome genes. These genes are turned on in the daytime in cells. They accumulate in the cytoplasm, and then they interact with each other and can dimerize, and then translocate back into the nucleus in the evening. As they accumulate in the nucleus, they then interact with clock and BMAL directly to inhibit their own transcription, to turn off their genes. Once the inhibitory complex turns over in the nucleus, then clock and BMAL become active again and start a new cycle the next morning. So this is a very simple view of the fundamental negative feedback loop in mammalian cells, in which there are 6 critical genes: clock, BMAL, PER1 and PER2, and Cry1 and Cry2. So this gene network has actually been extended by Louis Potocek's lab at UCSF to humans, in which individuals having a sleep disorder called advanced sleep phase syndrome, which is a timing of sleep disorder, in this very large kindred that they isolated, they were able to show that this dominant mutation was caused by a missense mutation in the human period 2 gene. They've subsequently shown 2 other examples of genes also in this gene network involved in advanced sleep phase syndrome and in short sleep in humans. And so we can extend this model of the clock all the way from Drosophila to mouse, and we presume to humans as well. So here's a more intermediate picture of the clock about 6 years ago, in which we have the clock BMAL transcriptional activators acting on PER1 and 2 and Cry1 and Cry2 to form this inhibitory complex. One level of post-transcriptional regulation of period is by the kinase, casein kinase 1 epsilon, which we also identified by positional cloning of a mutation in the golden hamster. I'm not going to tell you about that today. But this kinase is also highly conserved in flies. The original double-time mutation was first isolated in Drosophila and acts in a very similar way to regulate the half-life or turnover of the period protein. And then more recently, Uli Shribler's lab has shown that the nuclear receptor, Rev-erba alpha, is under direct clock control. And Rev-erba is a very strong repressor of BMAL1 transcription and clock transcription in most tissues. And this leads to a second oscillation in the level of BMAL and clock. This second loop is not essential. It can be interrupted and the primary loop still functions. And so this primary loop is what we consider the core mechanism of the clock. Now, is this sort of the end of the story? And the answer is no. We have lots of reasons to think that there are many other genes involved in this pathway. So we have additional mouse mutants that are being cloned. And I'll show you one example that we cloned about 3 years ago. But one of the large projects undertaken by us at Northwestern and funded by the NIH in around 2001, 2002 was a very large-scale ENU mutagenesis screen in mice for many behavioral phenotypes beyond circadian rhythms. We focused on 5 major domains: circadian rhythms, fear conditioning, vision screen, a metabolic, obesity, and motor screen, and then a psychostimulant response screen. And I don't have time to tell you about this screen, but over that 5-year period, we isolated over 100 new mutations, and currently about 20 of them have been positionally cloned, but we have yet another long list of mutants in the pipeline for identification. In the case of circadian mutants, half of the new mutations that we've cloned are novel, and the other half have been re-mutations in known core clock genes. So the first example of a new mutation that came out of this screen, which is shown here, which is a 3-generation recessive screen, is this mouse, which has a 26-hour clock as compared to normal mice. And this mutation we called overtime is about 10 standard deviations away from the mean, which is shown here for a much larger set of mice here, about 3,000 mice in this particular screen here. This mutation is also semi-dominant. It's heterozygotes have a slightly longer period, and then homozygotes have this 26-hour period. And you can see the histograms of the period values in a large mapping cross that we used to map the mutation. And this was interesting because it mapped the chromosome 14 on the distal end, and there were no known clock genes there. And so this was the ultimate region. It's about 4 megabases and it had about 18 genes. We sequenced all of these genes, and there was only a single point mutation in this gene here, FBXL3, which was a missense mutation in the C-terminus. And FBXL3 is interesting because it was a previously orphan member of the F-box protein family, which is an F-box protein with leucine-rich repeats. The founding member of this family is called SCIP2, very important E3 ligase for the cell cycle. And ultimately, we and 2 other laboratories that incredibly found this gene at the same time showed that FBXL3 interacts with the cryptochrome proteins to stabilize their degradation. And so these are MEFs isolated from either wild-type mice or overtime homozygous mutant mice, looking at the half-life of native Cry, after cyclohexamide treatment. And you see that native Cry has a half-life of about 5 hours, but in the overtime mutant, Cry is stabilized. The half-life of PER, on the other hand, is not influenced. And so, to make a long story short, FBXL3 forms a new E3 ligase complex that interacts selectively with the cryptochrome 1 and 2 proteins to target them for ubiquitination and then ultimately degradation by the 26S proteasome. And loss of function mutations in this gene lead to a chronic repressive state of the clock and a prolongation of the turnover of the cryptochrome protein, which can then cause the period length to be lengthened. So this is looking at just the core loop, where what we see is that the degradation of PER and Cry, as well as the transcription of PER and Cry, are regulated on a circadian basis. And at least the degradation side, or the half-life regulation of the PER and Cry proteins, contributes significantly to the actual period length of the system. Okay, so how do we study this system? So one of the things we do is we take genes like PER, or PER1 or PER2 that are cycling, and such as this. So this is showing you PER2 by in situ hybridization in the suprachiasmatic nucleus of an SCN at the peak or the trough in a normal mouse, or in this case, a BMAL null mutation, which completely flattens PER2. We use PER2 as a marker and couple it with luciferase. So one of our best reporters is a knock-in mouse where we knock in luciferase in frame into the C-terminus of the PER2 locus by gene targeting and replace the native gene with a PER2 fusion. Okay? This allows us to then visualize PER protein abundance by bioluminescence. And so this movie here shows you a slice, organotypic slice through the SCN in which you can see a wave of bioluminescence going across the nucleus over a 7-day experiment, in this case done by David Welsh at the Scripps and now at UCSD. If you look at different tissues in the mouse, essentially any tissue that you can keep alive as an explant can express a circadian rhythm. So this mouse revealed that many tissues and organ systems throughout the body have circadian oscillations. So the liver, pituitary gland, kidney, lung, or even the tip of the tail. And so if you prepare fibroblasts from this mouse, grow them to confluence, and then use imaging, as shown in this time-lapse movie, which is actually a 6-week-long experiment, you can measure circadian rhythms at the single-cell level that persists for 42 days in culture. So those are 6 examples of cells taken from this experiment. So this has completely changed our view of clock systems. We actually had no idea that the lowly fibroblast had such a robust circadian clock. Macroscopically, these cultures, the rhythms, the cells and the rhythms are desynchronized, and so you have to use a synchronizing pulse to see the rhythm macroscopically. But using single-cell imaging, you can see that the rhythm is actually much stronger at the single-cell level, and that the desynchronization of the rhythm and damping of the macroscopic rhythm is really due to loss of synchrony. And so if we look in more detail, here's a heat map representation of 75 fibroblasts taken from a single field of view. Initially, they're somewhat synchronized by a medium change here, their phases. But over time, over that week's time, the period differences among these cells leads to complete uncoupling of their phases. And as you can see, they're randomly distributed at all phases. One of the interesting features is that the period values for individual cells is much broader than what we see at the organismal level. So single cells have periods that go from 22 hours up to 30 hours. But the average is about 24 or 25 hours. Okay? And so one of the curious or interesting questions that we don't understand today is what makes a cell have a short period and another cell have a long period. These appear to be relatively stable over time. And we really don't understand what it is that gives a single cell its intrinsic period. But overall, the average of those cells is very close to what we see at the tissue or organismal level. Okay? So given that clocks may exist throughout the body in many different cell types, one of the questions we went back to ask is, what are the effects of the so-called classic clock mutations, which are isolated at the behavioral level. What are the effects of those mutations on these cellular clocks? And so, for example, if we take the cryptochrome 1 and 2 loss of function mutations, a Cry1 knockout mouse has a short period, Cry2 knockout mouse has a long period, and the double knockout has a loss of rhythm phenotype. So there are two paralogs, you have to knock out both to get a loss of rhythm phenotype. But the individual knockouts have these interesting period phenotypes, which we actually don't understand today. If we then use per-luck reporting as an output, we can ask, what effect does Cry or per loss of function mutation, what do these have on SCN organotypic explants or peripheral tissue explants, such as the lung in this case? And what we see is in the SCN, the effects of these mutations are very similar to what we see at the organismal or behavioral level. They're very subtle, and they're in the same direction. Per1 loss of function does almost nothing to the SCN or to behavior. Same with per3. Cry1 shortens the period, Cry2 lengthens the period, and the double knockout leads to loss of rhythm. Okay? But in the lung, we see a different picture. Per1 alone causes this very strong loss of rhythm phenotype, as does Cry1 alone. But this is selective or specific because per3 or Cry2, for example, don't have these effects. Okay? So what is the cause of this? Is it because the SCN is different from the lung? Or is it because the SCN is a neural network and maybe the lung isn't? And so we asked, is the effect of the mutation cell autonomous? So if we dissociate fibroblasts or SCN neurons, not explants, but dissociated neurons, and then look at the single cell phenotype, what we see is the effects of these mutations are cell autonomous. Cry1 causes a loss of rhythm phenotype in both the fibroblast and the SCN. Same with per1. And so it is not the case that the SCN cell is somehow different than a fibroblast as far as the clock is concerned. At the cell autonomous level, the isolated cell reveals this sensitivity to subtle mutation, which we cannot see at the SCN network level. So if we look at a Cry2 knockout, either in a slice or in dissociated cells, we see in the slice at the single cell level using imaging techniques, these are 20 SCN neurons represented as a heat map, we see that they're coherent and synchronized. But when they're isolated or dissociated, each neuron has a rhythm, but they're no longer synchronized. This looks like the picture I showed you for fibroblasts before. Okay? They oscillate, but they don't communicate. If we then look at Cry1 slices, we see that the slice is rhythmic. The cells are rhythmic and coherent, but there's an interesting feature. The phases are not progressing as a wave. They're all synchronized. These bars are vertical because they're aligned in time. They're occurring at the same time. So the nucleus is flashing on and off at the same time, rather than having this wave that progresses over the nucleus. And if you look at dissociated single cells, each cell fails to generate coherent circadian oscillations. And thus, it seems likely there's something about the organization of the SCN that can rescue this defect or overcome this defect. And so using modeling, modeling done by Frank Doyle at UC Santa Barbara, if you model very weak oscillators that cannot generate self-sustained oscillations over time, shown here as a heat map, and all you do is you couple them together, then the population then can couple together, synchronize among one another, and produce coherent oscillations. So coupling alone can account for such a rescue that we see. And so this set of work, I think, has been important because most of our analysis of circadian rhythms has been focused on circadian behavioral outputs and the suprachiasmatic nucleus. We've always been very neural-centric about this. But what we now realize is that the SCN circuitry actually can transform the cell-autonomous output of these clocks. And in particular, the coupling can actually rescue a genetic defect at the cell-autonomous level and make the rhythm actually appear relatively normal when at the cell level it's very abnormal. In peripheral tissues, where the coupling is much weaker, this rescue is not obvious, and so we see the cell-autonomous defect directly, such as in the lung. And when there isn't a cell-autonomous defect, such as in Cry2 mutant cells, then peripheral tissues still generate very strong oscillations. So we think that this kind of model can account for the differences we see between the SCN and peripheral tissues in the strength of the rhythms that are expressed and their differential sensitivity to subtle loss of function mutations. The other reason this is important is that if we're trying to understand the function of Cry1 or Cry2, those effects of Cry1 are actually masked by the SCN. And so if we're trying to understand the biochemical function of Cry1, for example, it might be better to look at the cell-autonomous or single-cell phenotype, where we see the direct effect of loss of Cry1 as compared to Cry2. Okay, so there's a similar story that occurred with the clock gene. So the mutation that I told you about was a dominant negative mutation. When the clock gene was knocked out by gene targeting, causing a null mutation, the mouse still had a circadian phenotype. So this very provocative title here, Clock Shock, by my friend Steve Rev-erba. And so this is really an illustration of the difference between an antimorphic mutation and a null mutation. It turns out that clock has a close paralog that's called NPAS2. When you look at either NPAS2 knockouts or clock knockouts at the behavioral level, they have very subtle phenotypes, almost no phenotype. But if you make the double knockout, clock NPAS2 double null, then there's a very strong loss of rhythm phenotype. So this is very similar to what I've just showed you for Cry1 and Cry2, and also what you see with PER1 and PER2. And in addition, we see this central versus peripheral differential sensitivity to mutation also. So these are wild-type liver or lung per-luck records. If you knock out NPAS2, this does nothing. But if you knock out clock, this causes a very strong peripheral loss of rhythm phenotype, as well as the double knockout, almost as strong. Okay? And so we can see a similar or analogous kind of picture here. Clock appears to cause a cell-autonomous defect, which we can see directly in peripheral tissues. But this defect can be rescued by the SCN so that circadian behavior looks relatively normal. In contrast, NPAS2 does not appear to have a cell-autonomous defect. It also does not cause a defect in peripheral tissues. And so we think that this is the reason that clock null mice still have behavioral rhythms. Okay, so how is it that we study this system? And I want to give you two examples of how we are studying the system. Currently, currently. Both of these examples come from an NIMH Conti Center for Neuroscience Research that I've had for the last 5 years at Northwestern before moving to Southwestern. And these are the individuals involved in the Conti Center: Mike Menaker, Steve McKnight, Carla Green, John Hoganesh, Louis Potacek, and Inwei Fu. And then David Furster and Mike Wellams, who are core leaders for both engineering and software development. So one of the ways that we have studied the system is to try to manipulate clock gene expression in a tissue-specific and conditional manner using the tetracycline transactivator system. And so this is a bi-transgenic system. You have a mouse that drives TTA, a synthetic bacterial activator made by Herman Bouchard. In this case, it's driven by secretagranin 2. This mouse was actually made by Jason Chong in my lab when he was a student. Jason is now here at NIH with Tom Wellams. You cross this mouse to a responder mouse, which has a TETO operator set of sequences driving the gene of interest, in this case PER2. And so when you have the double transgenic mice, you get high expression in the brain as compared to controls. And this is just showing you PER2 endogenous expression in these controls here, and then overexpression in the double transgenic mice. But you get high SCN expression, about 3x over wild type. Okay? And so here are the behavioral records of these mice. This is a wild-type, non-transgenic mouse, but a litter mate. The yellow shows when the mouse has doxycycline in the drinking water. There's no effect in this mouse. These are our two independent TETO PER2 responder lines, which are also normal. Okay? Here's the secretagranin 2 TTA driver mouse. It's also normal. And then in the double transgenic mice, showing you two independent responder lines, we get a very strong loss of rhythm phenotype by constitutive expression of PER2 in the brain and SCN. In both of these animals, in both lines. And when you give the animal doxycycline to turn off that PER2 expression, then the rhythm comes back and it looks normal. So this is conditional and reverse. And then if we then shift the animal back to water, which turns on the transgene again, then the animal goes arrhythmic again. So it's tissue-specific, conditional, and reversible. So how is PER2 affecting the clock system? And this is, as I showed you before, this is working in the whole animal. So it has to be a strong effect because the SCN can rescue subtle effects. Okay? And so in collaboration with Shugon Lee, we've used cell-based and in vivo systems to understand at a molecular level what PER2 overexpression might be doing. And so this is the PER2 luck rhythm from fibroblasts isolated from these mice in macroscopic cultures. Okay? And this is a Western blot of PER in the liver, the cycle over the time of day. It cycles both in abundance and in phosphorylation state. So you see this slower migrating species of PER as it accumulates. In the PER2 luck fusion protein in the liver. And also we see a similar pattern in fibroblasts from these PER2 luck mice as well. Okay? So using adenovirus, we constitutively expressed Cry1, PER1, or PER2 in these fibroblasts. And what you see is the GFP control has no effect compared to no virus. Surprisingly, Cry1 overexpression also has no apparent effect, but constitutive expression of PER1 or PER2 strongly suppresses the rhythm. Okay? And if we look, so this is just showing you so this is a PER2 western in the fibroblasts infected with adeno. This is the PER1 level in the GFP control cryoverexpression or the PER2. And what you see is in PER1 there's this oscillation, okay, in both GFP control and Cry expression, but it's constitutively high and non-rhythmic in the PER2 overexpression. Similar thing happens here. This is PER2, it's cycling, and it's flattened by PER2 overexpression exogenously. At the RNA level, this leads to changes in the expression of these various clock genes. So Cry1 is actually overexpressed. This is sort of a funny result. But two target genes of clock and BMAL are repressed by overexpression of PER2, Rev-erba alpha and DBP, shown in red here. Okay. Those were in fibroblasts. We also could inject the virus into the tail vein of mice and infect the liver very efficiently. So in mice, we see very nice PER oscillation or PER2 oscillation, and overexpression of PER2 also has the same effect. So if we look at RNAs for PER1 and PER2 Cry1, we see that PER2 constitutive expression flattens these rhythms. Same with DBP and Rev-erba alpha. So these are all clock BMAL EBOX target genes, as if the system is constitutively repressed by PER2 overexpression. This is a very complicated set of IPs, but what we're doing is looking at the interaction of clock or BMAL by IP and seeing how it interacts with PER or Cry1 or Cry2, okay? And we're looking at 2 times CT8 or CT16. So what you see in a normal mouse is there's a rhythmic interaction of Cry1 and Cry2 with clock. No interaction here at 8, strong interaction at 16. Same thing with BMAL. GFP or Cry1 overexpression does nothing to this rhythmic interaction, but PER2 overexpression causes constitutive interaction of Cry1 and Cry2 and PER with clock and BMAL at both times. Okay? To see if PER is necessary for this interaction, we use PER1-2 null fibroblasts. And you can do the same experiment. In wild type, you see this rhythmic interaction of Cry1 with clock and BMAL at the 2 times 8, no interaction, lots of interaction. And in the absence of PER, what you see is that Cry does not interact with either clock or BMAL. So it appears that Cry requires PER in order to interact with clock and BMAL. If we use in vitro translated proteins to try to do a stoichiometric experiment and ask which protein, PER or Cry, binds more efficiently to clock and BMAL, what we see is that PER2 binds stronger than Cry1 to both clock and BMAL under these conditions. And if we add PER2 to this mix, then this facilitates the interaction of Cry1 with both clock and BMAL, this comparison down here. Okay? So this suggests that PER is required for Cry interaction with clock BMAL. And because PER is normally rate limiting, that may be why Cry doesn't have an effect when we overexpress it. So to address the rhythmic aspect of PER expression, what we did was to drive PER2 with a rhythmic promoter, but in this case, an inappropriate promoter, that's BMAL, B-mal expression is opposite to the phase of PER2. So we're expressing PER2 in antiphase, at the wrong phase, but we're overexpressing it. And what this does is that this leads to a loss of rhythm. So you have to have PER2 cycling, but it also has to cycle in the right phase. So if you then drive PER2 with its own promoter, you can get an overexpression of PER2, but the cycle continues. So the effect of PER in abolishing the rhythm is not just the level of expression, but it's whether it's cycling or not, and whether it's the appropriate phase of cycling or not. Okay? So in the normal negative feedback loop, what we think is going on is that Cry is in large excess in the cytoplasm. And Shugon Lee has shown previously that PER is required for translocation of Cry into the nucleus. Our new experiment suggests that PER is actually required for the interaction of Cry with clock and B-mal. And this is a surprise. Most people have thought that Cry is the major repressor in the system, not PER. And so it was surprising that overexpression of Cry had almost no effect. When we constitutively overexpress PER, this leads to a chronic state of negative feedback by Cry and PER on clock and B-mal, which downregulates many cycling target genes and leads to this loss of rhythm phenotype. Okay, and so in the last few minutes, I want to just quickly show a second project from this Conti Center, which addresses a potential problem that we have in humans, and that is that we respond, of course, to the natural daylight cycle, but we also regulate our own light cycles ourselves. And, of course, we also travel long distances transmeridianly. And this has led to a number of problems that evolution did not anticipate. And so if you take a rat model and fly it to Paris, advance its clock by 6 hours, and you monitor the rhythms in the SCN and various organ systems in the body, what happens is the SCN can actually reset pretty fast. Your brain, at least in the rat, can reset in almost 1 day. But the peripheral rhythms are very slow. In this case, they took 6 days to shift over in experiments done by Shin Yamazaki at the University of Virginia. And so this idea that we have clocks not only in the brain but in the body has led to, I think, a new set of targets for intervention of the clock system. Previously, we would have thought, well, you have to reset your brain and then everything is fine. The body will just follow. But this kind of experiment shows that even though the brain is reset, the body is still very sluggish to respond. And so another project within this NIMH Conti Center with Steve McKnight was to develop a cell-based high-throughput screen for rhythms using fibroblasts. And so in 384-well format, shown here, this is our viewer, we can have individual wells. The blue traces show DMSO control wells here, and then the purple or pink shows a positive control. It's a kinase inhibitor that delays the rhythm. And then the other wells shown in yellow can then just have experimental compounds. And then in this software interface, it automatically extracts the period value and the phase of the rhythm from all these experimental wells and plots them here. And so you can see this is the cluster of experimental wells in this particular experiment. These are the positive controls. And so using this kind of system, Jake Chen at UT Southwestern was able to screen a 200,000 compound library at UT Southwestern in the Department of Biochemistry, created by Steve McKnight. He then validated the initial hits, and then he did secondary screening with a different reporter line, a human cell line. Then he did further validation on HER2-LUC explant tissue, the SCN and pituitary. And then he also asked whether these compounds might rescue mutant clock cells. Now, in the screen, what we found is that many compounds can lead to a reduction in amplitude and a lengthening of period. And we consider many of those effects nonspecific, so metabolic inhibitors or anything detrimental to the cell reduces the amplitude. And it's easy to lengthen the period. So we focused on compounds that could increase amplitude and shorten period because a priori it's harder to do that in a nonspecific way, okay? And so these are the 24 initial hits from this screen after validation. I'm sure you can't see this, but what we found is some compounds that lengthen, which are these three, but we focused primarily on compounds that enhance amplitude, which are the majority of these other compounds here. We also rescreened them on this human U2OS cell, so we have two different cell lines. And what we found are a set of compounds that consistently shorten the period and enhance amplitude in both mouse and human cells. And interestingly, these compounds can actually enhance amplitude in mutant fibroblasts. So clock mutant fibroblasts or BMAL heterozygous knockout fibroblasts all have very strong reductions in amplitude. And these compounds can actually rescue this defect. In particular, compound 17 here can completely rescue the mutant reduction in the amplitude here in a dose-dependent way. And so Jake was able to screen this library, got about 500 hits, validated about 50 of them, and then focused on about 24 that worked in both mouse and human cells. And he's been able to rescue or find at least one or two compounds that appear to rescue mutant clocks. And so we think that this might be an interesting approach to deal with potential circadian types of deficits. So normally our system is synchronized, wake, feeding, and energy utilization in the body. But under many conditions, such as shift work or other kinds of disruptions, our system can actually be desynchronized. There are animal model examples showing that this can lead to some types of metabolic phenotypes and ultimately pathological and molecular changes in many different pathways. And so one idea is that perhaps small molecules could be used as an agent to synchronize and enhance the amplitude of these clocks and perhaps be beneficial in normalizing rhythms. So I'll stop there. A number of people contributed to this work. I mentioned most of them, but I should point out Sung-Hee Yu, who made the HER2-LUC mouse, Aaron Shermer, who made the HER2 overexpression mouse, and then very important collaborations with Jake Chen, Steve McKnight at UT Southwestern, and Shugon Lee and Rongmin Chen at Florida State University. Thank you very much.
We have a few minutes, so if anyone has any questions they'd like to ask, please go to the mic. Looks like there's a few over there.
Is there a role of clock genes in peripheral organs independently of the SCN that might predispose to certain disease states?
Yes, we think that, so I didn't show any examples of this, but there's a beautiful experiment done by Uli Schibler's lab where we had an inducible system to shut off the clock in the liver, but the rest of the mouse was normal. And in the liver, if you do a microarray profiling experiment, you will find many hundreds of genes that are cycling in the liver. And when they turned off the clock in the liver, the majority of those cycling genes were flattened. And so that suggested that for, say, 90% of the cycling genes in the liver, the cell-autonomous local clock was the predominant regulator of that cycling transcription. But there are also central inputs into that liver, and that was the purpose of their original experiment. And so they found a smaller set of genes that actually respond to systemic cues. And those are interesting. One of those is PER2, actually, which is a clock gene itself, but it responds to systemic cues. And then other ones are members of the heat shock family, actually.
And those changes might predispose, you might think, based on those particular genes, that might predispose to certain diseases, for example?
Yeah, there are many tissue-specific targets of clock and BMAL in, say, the liver or any other tissue. They're downstream of clock. And so if clock is mutant in that tissue, then many different targets and their expression is altered.
You mentioned about all the organs. How about the heart? Is it similar to skeletal muscle?
Yeah, so we haven't done very much work on the heart, but for example, you can do a Perluck experiment on the heart, and it also oscillates. So it does have the capacity to oscillate.
And for the drugs to work, do they have to cross blood-brain barrier to affect the SCN cells?
Yeah, so that's one thing, is we have not tested any of these compounds for central action. But because many of the cells are going to be peripheral targets, it may be okay to have drugs that are actually only acting peripherally.
So when people get old, it appears that the sleep cycle phase shifts, and perhaps even the amplitude may dampen in that sleep is poor and there's more napping during the day. What is known about aging and the circadian rhythm, both in amplitude and phase shift?
Yeah, so in general, there's evidence at the molecular level, and Aaron was involved in some of these experiments, I think, that the amplitude of rhythms of clock gene expression are lowered with aging. If you look at sleep, you do get fragmentation and less efficient sleep, and lower efficiency of sleep in humans also. So there are many quantitative changes. They're all reductions that occur with aging. But I don't think anybody yet has pinpointed a pathway or a mechanism to explain that. Would that be fair?
That's true.
Yeah.
That's true. We'll take the two remaining questions, but I just wanted to point out that after this, there's a reception next door over there. So if you want to meet Dr. Takahashi or talk more with him, please come over to the reception. So let's go. We're there first, and then we'll...
Thank you. I would like to ask you about the other end of the scale, about embryonic development. So what I have read a couple of years ago, that the central clock gets activated only after birth. What about the cell and autonomous clock?
Yeah, that's a very good question. So it's actually been difficult to address that question. If you look at the ovary with Perluck, the ovary has a circadian rhythm. And there's some evidence that even oocytes can have a rhythm. But during embryogenesis, no one has yet been able to measure per-luck rhythms reliably. It's hard to culture embryos, that's really why. And so a few days before birth, you can see clock gene expression and rhythms appearing. So that's clear, but what's happening in between is not clear. Okay? The other piece of evidence I can give you is that embryonic stem cells in general do not have rhythms. But if you differentiate them, then they acquire rhythms. So that's another way of looking at that.
So I have a question about FBXL3.
Mm-hmm.
So is there any evidence that its stability or activity of that E3 ligase is itself regulated by any cellular inputs? Does it have a small molecule binding domain on it?
Yeah, that's a very good question. So there's a recent paper from Ron Evans' lab showing that AMP kinase, which is thought to be a metabolically regulated kinase, phosphorylates two sites on the cryptochrome protein, and those phosphorylation sites are involved in the regulation of FBXL3 interaction with Cry. But so far, there isn't any, I'm not aware of any information on FBXL3 itself.
Thank you.
Mm-hmm. Thank you.
Thanks, Joe.